დავიწყოთ მე მგონი არა 124 არის უკვე. და ვინ ნაწილი შემოგვიერთდება მე. არ მიკროფონი გამორთული გაქვს.
დიახ, ერთი წუთით დავველოდოთ, ხალბატონ ლეილა შემოგვიერთდება და დავიწყებთ.
ბატონო ზაალ, მე მგონი ჯობია დავიწყოთ. ვინ შემოგვიერთდება, შემოგვიერთდება, რომ აღარ მოგაცინოთ.
არა, უბრალოდ მე ერთმანეთზე სხვა ჩართვა მაქვს.
კი ბატონო, რომ არ მოგაცინოთ, ჯობია დავიწყოთ მგონია.
Gadzgent თუ შეიძლება თუ ნებას მომხდართ. Greetings everyone. Thank you very much for joining this meeting. This is Elizabeth from International Relations Department. Let me introduce today's meeting speaker, Mr. Zal Kokaya, who is a doctor of biological sciences and professor of experimental medicine research at Lund University. Within a lecture, we will focus on the topic cell reprogramming future potential treatments for stroke patients. I wish you a successful and fruitful meeting, and thank you for your attention.
All right. Good morning. We have morning still here in Sweden. It is a pleasure to join you and to give a little bit of talk. So my talk will be consisting of two parts. First, I will introduce the topic of stroke and the stem cells. And then in the second part of my talk, I will tell you a little bit about our research and how we see the development of this research, which will be ultimately and hopefully translated into the clinical settings. So cell reprogramming future potential for stroke patients. Let's start. What do we know about brain? So I understand there are lots of medical people around, but still let's remember what we know about this human brain. It's not that big. It's just 1.3, 1.5 kilos in weight. Mostly it's water, surprisingly, and some other components. But what is important that despite the fact that it weighs only 2% of the body weight, it consumes a lot of blood, which means that energetically it's very demanding and functionally it has a very big role in the operating the whole body. So if we look on the blood flow is necessary, of course, to provide the organs, including brain, with oxygen and the glucose. But sometimes the vessels which are providing blood to the brain might get disrupted, and this destruction might be due to the occlusion of the part of the body, quite often due to arrhythmia. It's coming from the heart, and due to this destruction, then the area of the brain will not get sufficient supply with the blood, becomes ischemic, and the fact that they don't have oxygen and glucose will lead to the damage and death of these cells, as shown here. There is another type of stroke, which is representing roughly 10% of all strokes, ischemia being the majority of cases, and that's due to the rupture of blood vessels caused by high blood pressure or some abnormalities in the vessel, like aneurysm, etc. But today we will talk mostly about ischemic stroke because it represents the vast majority of all stroke cases. So it's a leading cause of the disability, and this is something we need to remember, that majority of the adult disability is caused by the stroke. And in parallel, it is third leading cause of death in the developed countries. It's about 3.5 million people in EU with 700 new cases every year. The cost of stroke is enormous. The survivors of the stroke require some treatment assistance, also the cost caused by the lack of their ability to work. Therefore, it's very high cost, and economically it's very devastating. So 2/3 of the patients will survive, and then they can have very different degree of the disability, starting with minor impairment up to the unconscious comatosic condition. So it's a very wide spectrum of the disabilities which one can observe depending on the size of the lesion and depending on the location of the lesion. What is also important to remember that the stroke is age-related disease, and the chances to get the stroke after age 55, it doubles. And therefore, with aging population, we need to come up with something, because we don't have basically any treatment in helping the recovery and the regeneration after lesion takes place. And you can see on this graph that with aging population, the increase of the stroke events, as well as the increase in the number of people living with stroke, it's quite substantial. So these are the phases of the stroke, starting from hyperacute, acute, subacute, and in the end, the chronic phase. And these phases are starting from hours, ending up to chronic stage when the patient will survive. Currently, we have very efficient treatment for the acute phase, hyperacute phase, within 5-6 hours after the onset of the stroke. And you can see here on this graph the occlusion of the vessels, and after thrombolysis, the clot opens and then reperfusion occurs, and it's very efficient treatment. But unfortunately, only 10% of patients could be suitable for this kind of treatment. So therefore, we need more treatments which will help us with already established lesion to regain the function and help the patients. What's happening in the brain once the ischemia occurs? Every hour that goes by without clinical intervention, you get 120 million neurons dying, 830 billion synaptic connections are lost, and 714 kilometers of myelinated fibers are damaged. So this is enormous events which is happening in the brain. And then you can see here, this is the brain neuronal network stained with different markers. And lots of events are occurring in response to this damage, starting from gene expression, dendritic spine turnover, rearrangement in the dendritic spines. And you know that the dendritic spines are the parts where the synaptic connections are established. Axonal sprouting, the axons will be rearranging their connections. There are lots of cell genesis, including neurogenesis, which we discovered post-stroke neurogenesis back in 2002. And of course, sensory and motor cortical rearrangement, representation changes. And all these events are directed to change and support to recover and regain the network, neuronal network. Similar to fishing nets, the old fishing nets, if it gets whole, if the network of the thread is destructed, then you will get hole in the net, and then function is impaired of the net to catch the fish. But you can repair it, you can restore the net with a new thread, and then function will be regained. I think this is a good parallel how we can maybe restore the network, neuronal network, which is damaged due to the stroke. In order to study the stroke, we have to have animal models. And here one example how one can induce stroke in animals and study it. That's what we do. You can put the fishing filament through the external carotid artery to the origin of the middle cerebral artery, and this is the artery which is most commonly occluded in the humans. And this occlusion, of course, will cause the damage, respective damage, and then you can withdraw this fiber and recirculation will take place. So the blood will be restored, blood flow. And depending on the extent of the duration of the occlusion, you can have defined striatal lesion as shown here, or you can have striatal and cortical lesion. However, you can have another model where you will occlude only distal branch of the middle cerebral artery, and that will ultimately cause only cortical lesion. And that's the model which we are using in our settings. So cortical stroke, and you see here in this red brain, this white shows the damage of the cortex and the rest of the brain, and it's always unilateral, of course, and the contralateral side will be affected of the brain. So in order to assess what happened with the animal, we have lots of different animal models, behavioral models to assess how they use the pose, how they behave as a response of this damage, because that's important to assess the degree of the damage, functionality of the damage. And if you want some treatment, also you should be able to assess the recovery, how well they regain the normal function. Therefore, we have a set of behavioral tests. After inducing stroke in animals, we can assess how much and what function has been impaired, as well as we can assess after treatment how much they regained this impaired function. And that's very important in experimental studies. Now, switching gears to what are the stem cells. So classical definition of the stem cell, that this is the primitive, very simple stem cell in a sense that it has only two functions. First function, to replicate itself, produce two identical daughter cells. And the second function, through the differentiation during the development or in the culture system, if you will apply certain substances and trophic factors, it could give rise to the specialized cells. It could be muscle cells, could be pancreatic beta cell, or could be fat cell, or in our case, of course, we are looking for the neuron. So please remember two functions, self-renewal and the differentiation potential. These are the two features of the stem cells. So stem cells are kind of originating from the developmental field, and everything starts for the fertilized egg. And this is totipotent stem cell fertilized egg, which means that it can give the rise origin to the whole live organism. Then the division of this fertilized egg starts, and then at the stage of the blastocysts, there is this inner cell mass, which is normally embryonic stem cell, what we call embryonic stem cell. And when people talk about stem cells, mostly they mean the embryonic stem cells. And they are pluripotent. And pluripotency means that they can generate any kind of the cells which are present in our body, more than 200 different types of the cells. They can generate, one can have these cells, hematopoietic stem cells, neural stem cells, mesenchymal stem cells, they can differentiate. And this becomes multipotent. This type of cells, multipotency, means that they can generate only the type of the tissue or cells which are specific for this organ system. Like mesenchymal stem cells can generate only fat, cartilage, and the bone or connective tissue. Neural stem cells have their own niche. So neural stem cells can self-renew because they are real stem cells, and they can generate all neural type of cells, which is neuron, astrocytes, oligodendrocytes. So if the cell can replicate and give rise to the identical cells, and if you differentiate these cells, give rise to neuronal lineage, neurons, astrocytes, and oligodendrocytes, we can call this type of cells neural stem cells. And this is very important cell for us, for our topic. So now self-renewal and multipotency could be achieved through the symmetric division, and multipotency through asymmetric division. And then once the neural stem cell divides, it gives the neural progenitors. and neuroprogenitor, neural, it's not neuronal, neural progenitors. Neural progenitors can give neuroblast, which can give rise to the neurons, and the neural progenitor can give the rise to the glioblast, which can give us astrocytes and oligodendrocytes. So this is the kind of scheme. This is the setting which we can observe in the development of cells, and how it happens that asymmetry, that one cell divides and gives two types of cells, that could happen due to the fact that one of the cells will appear in the different environmental conditions, and then different factors, external factors, will influence the fate of these cells. Or it could be during the division that the inner content of the cells is not equally distributed, and then that could be also the mechanism of giving the differentiation of the stem cells. And here you can see that this NAMB protein, which is not equally distributed between the daughter cells, will lead to the fact that one of them will start the differentiation fate. Okay, so there is a mechanism which ensures the properties of the stem cells, which is again self-renewal capacity and the differentiation or multipotency capacity. Now, we have stem cells in many organs. However, most commonly and most known are the bone-derived stem cells, and all you know that this is the hematopoietic stem cells, which gives rise constantly throughout whole life. Our life depends on the work of the hematopoietic stem cells, because we need constant refreshment of our blood cells and immune cells, which are produced by these cells. But what is also important that we have in the brain, even in the adult brain, two areas. This is subventricular zone and the hippocampus, where we have the adult stem cells. And that's the good news, because before we thought that there is no way that you have production of new neurons in the brain. However, today we know that the adult brain has the capacity not only produce glial cells, but also produce new neurons. So that's important to remember. Now, during the development, this is a classical Weddington landscape. During the normal development, this stem cell decides which way to go, and depending which pathway this ball will select, it will become different type of neurons. However, not long ago, a little bit more than 10 years ago, it was discovered that you can reverse this process. From the differentiated cell, you can go back and produce the stem cell. And that was the Nobel Prize winner in 2012 by Yamanaka, and it's called induced pluripotent stem cells. So he took the skin cell, overexpressed 4 different transcription factors, and then he got back differentiated fibroblasts, differentiated skin cells, got back to the pluripotency. And then from there, you can generate that any kind of the tissue similar to what you can do with the embryonic stem cell. And that was reprogramming, which is a really important term. Moreover, now we know that without going back to pluripotency, with different set of transcription factors, we can directly transdifferentiate one postmitotic cell into another type of postmitotic cells. And that's what we do in my lab and many other labs nowadays. We take skin cells or blood cells and transfer them with the viruses, with different genes, and they become, and that's kind of science fiction, but that's reality. I didn't believe myself until I saw it with my eyes under the microscope, that the fibroblasts in 2 weeks give very nice rise of the neurons. And these are the functional neurons with action potentials and the synaptic connections, which is of course very important. Now, what can we do with this type of knowledge? So what we can do, we can generate from skin cells directly pluripotent stem cells, and from there we can develop neural stem cells and then manipulate in the culture system, label them. Now with the CRISPR approach, we can cut off some genes, we can insert in other genes, so we can do some manipulation and produce the type of the cells we want. And then these cells can be transplanted to the brain to replace dead or damaged neurons. And that's the philosophy which my lab is pursuing. And then these newly transplanted neurons can have different functions. They can enhance neuronal plasticity. I told you in the beginning of the lecture that there are lots of changes of the plasticities which are directed towards reorganization of the neuronal network. And with adding these new neurons, young neurons, non-committed neurons, we can enhance the capacity of the brain to rearrange, recover, self-cure, so to say, of the brain. So that's the good news, and also not only plasticity but the network. Network means that the neurons which are transplanted in the brain, in order to be part of the network, they should receive the connections and they should send the connection. So that's the whole function of the neuron, right? You know, so the neuron is communicator, passing the signals, passing the information. So it should be recipient of afferent information, receiving from other neurons, and it should be efferent. It should be transducer of the information. It should send the information in the forms of the action potential through the synapses. So afferent and efferent connections, unless they are established with the neuron will not be part of the network, okay? So, keeping that in mind, and you can see here the big hole in the cortex. This is the brain of the stroke patients. So our main task here, that with these stem cells, we can prove that we can replace dead or damaged neurons and restore neuronal network. And now I would like to show you some studies which can help us to prove that this could be the case in the experimental setting, and that could be the way to develop future therapies for stroke patients. Now, what we decided some years ago, generate not any kind of the neurons, but very specifically cortical type of the neurons, because majority of the stroke lesions and the most severe cases, they involve cortical lesion. And the cortex is very important. Also sensory function, speech, motor movements, etc. Executive function, of course, the cognitive functions like decision making, the responsibility taking, etc., discrimination, color, hearing, all this comes from the cortex, right? So therefore, we decided to make specific type of neurons, not any kind of generic neuron, but the cortical neurons. So what we did some years ago, we took the somatic cells, skin. You simply take the skin biopsy. You kind of like you do when you do testing, for instance, for melanoma skin sample. So you just, with a special device which has the needle, you just punch the skin. And then from the skin, you can isolate fibroblasts. And then from the fibroblasts, through the very complicated procedures with gene expression, with providing some small molecules, providing some growth factors, you can generate, and this is a complicated process, you can generate induced pluripotent stem cells. And that the delivery of these gene viruses could be the ones which incorporate, but nowadays we have also viruses which will not incorporate into the genome, Sendai viruses, or you can, with small molecules and the RNA delivery directly, you can produce the similar induced pluripotent stem cells. So induced pluripotent stem cells, then we can drive towards the neural line so that they will become neural stem cell line, which gives rise to the neurons, oligodendrocytes, and the astrocytes. And then as the next step, we can add some substances, some components, and produce cortical projection neurons. So this is the kind of schematic representation what we up to and how we work in our lab to generate cortical neurons starting from the human skins or human blood cells. Ultimately, these neurons we want to use for transplantation, for delivering into the brain, and for the integration of them into the brain and improvement of the impaired brain function. Now, what is happening under the normal condition, I would like to introduce engram cell concept in the stroke. When you do, and it comes from the memory setting. So engram is an experience which activates the population, certain set of the neurons that undergo persistent chemical and some structural changes and become engram cells. So when the similar stimulation comes, this set of the neurons will be active. And that's how memory works. So this engram comes from that. Similar happens when you have the motor actions or motor function. So the cells, certain cell set of neurons are involved in certain function. So when you get the stroke, certain given engram for the movement of the paw, for instance, as shown here, could be distracted, and the damaged motor engram will occur. Then with the time, due to this plasticity, which I told you, and coming back all the time, new arrangement, new connections will be established, and somewhat the recovery will occur. The function can be regained, and that could be varying quite a bit from minor to major. So the idea which we have, this spontaneous recovery, spontaneous regeneration, to promote it, that by delivering extra neurons, kind of building materials for rearrangement of this network, we would like to enhance this spontaneously occurring process and help body, help the brain to regain function more efficiently. And that's the whole philosophy, whole idea behind all the work which I will be presenting now. Is that understood? Is that clear now? I hope so. If not, then we can discuss a little bit more in the question and answer session. Now, here you see the complicated setting, how we, from the induced pluripotence system, how we generate neurons. And then you can see what you see in the culture, in the phase contrast, in these black and white panels, how the cells look and how their transformation occurs. And then below you see the staining with different antibodies, then fluorescent tags, which you can stain, fix the cells at different stages, stain them, and then identify their morphology or identify their phenotype, to which class of the neurons they belong, etc. So that has been published quite some time ago, almost 10 years ago, in the journal called Brain, and we can claim here that human-induced pluripotent stem cell-derived cortical neurons integrate in stroke-injured cortex and improve functional recovery. So here we showed that with the addition of certain molecules like BMP, bone morphometric protein 4, and Wnt3 transcription factor and cyclopamine, we can obtain the neurons with specific phenotype, with specific properties. They express transcription factors which are characteristic for the cortical neurons. They have their appearance are quite neuronal, and they are glutamatergic, because glutamate as a transmitter, this is the major transmitter for the cortical projection neurons. And what is also important, that they have these transcription factors like SERV2 and BRAIN2, and these are the hallmark for the transcription for the cortical projection neurons. So the summary is that we can make the neurons which have the cortical gene expression, pyramidal morphology, and what is also very important, electrophysiologically, they can generate action potentials, and they have the ion channels, and that's the hallmark of neurons being the neuron. But the question was still, after that study, that can human skin-derived neurons receive synaptic connections from the host? And then we continued, and in the science in general, it's very important to be consistent. So you take the research line and continue and expand and go deeper, and more you do, more questions arise. And this is the question. Okay, we get morphologically, genetically, based on the gene expression, cortical neurons. are they really functional in the sense that they can get the connections from the host? We knew that if you will stimulate outside if this is the transplant and you stimulate the cortex of the host, you can get the response, and this response can be blocked by the blockers of glutamine. But we didn't know whether this was synaptic direct connections from the host to the transplanted neurons, or it was inside the transplant. So what we designed a study with lots of, I will not go into technical details. We use multiple, and modern science is multidisciplinary. You have to use many different techniques and technologies in order to have successful study and successful publication. That's the only way to be successful nowadays in any field of the science, including the biomedical sciences. So we use electron microscopy. This is very old and classical method. It's not used very often nowadays, but if you use it in purpose, it could be very powerful. So you can see here with the electron microscopy, there is a synapse, and the synapse consists of postsynapse and the presynapse. And if our grafted cells, which are in green here indicated, have the postsynapse, which can be identified, that means that there is the synaptic connection coming to our neurons. That was good news. But then we switched to another technology, which is a very strong and very interesting technology. It's rabies virus, and you all heard about the rabies virus. But this is the avian rabies, bird rabies, which cannot infect humans. It's species specific. So what we did, we took our neurons generated in the culture system and introduced the receptor for this avian rabies, and also with the viruses. And also we introduced the glycoprotein, and glycoprotein is very important in order for the rabies virus through the synapse to move from one neuron to another. And you know that the rabies virus affects the brain and kills the neurons, but it spreads in the brain in a way that it goes from one neuron to another through the synapse. So what we did, so we tagged it with this GFP, with green fluorescent protein. We introduced the receptor, and we introduced the G protein, which allows this virus, once it gets in the neurons through the synapse, to get infection to another cell. When we transplanted such neurons into the rat brain, and these are human neurons, we injected the rabies virus. And when you inject rabies virus, it needs receptor, and receptor is only on grafted neurons, because this avian rabies receptor is not existing in the mammalian, in the rat brain. So the rabies virus was infecting only our grafted neurons, because they were having only ones who had the receptor. Okay, that's fine. And then because this rabies virus was tagged with the fluorescent protein, it turned into red cells. All right, because rabies was red. Now, we'll get it, because this rabies virus, this neuron also has this G protein, and this will also turn red, but it will be only red. But our original cell, I told you, it had GFP, green fluorescent protein, and with rabies virus, it got also red fluorescent protein. So double colored, red and green, is our grafted neuron infected with rabies virus, but all the neurons which make synapse connections with these grafted neurons will become only red. So we can discriminate now with the microscopy and with immunohistochemistry the difference between those two populations of the neurons. But other cells will not get infections because they don't have any glycoprotein. So here we are. So this is grafted area, grafted neurons. And these neurons around, only red ones, these are the ones which made synapse with our grafted neurons. And they taken out the rabies virus and turned into the red. This is the thalamus. So the graft is in the cortex, and we get stained cells in the thalamus. Why does it happen? Because thalamus has very big projections to the cortex. All sensory information from the periphery, somatosensory information, to our cortex comes through the thalamus. So the thalamus-derived neurons, they make synapse with grafted neurons, take out the rabies virus and turn into rat. And thalamus is quite far away from the cortex. This is, again, adjacent areas of the cortex. And what was very important, so the corpus callosum, and this is the structure where axons, connections from one side of the brain goes to another. You know that the two sides of the brain, and to our big surprise, on the contralateral side, so you have damage on one side, you have transplant in this side, and then over the sudden you get the stainings on the contralateral side. The only way this to happen, that this contralateral side sends the axons to the damaged side, makes connection with grafted neurons, because these are the only one which gets the rabies virus, and takes it out and then gets stained on the contralateral side. That was, this is, look at this beautiful neuron. And here you can see on the schematics, this black is the stroke lesion, green is the transplant, and the cells which you see on the screen is depicted on the other side of the brain, far away. And that's because this neuron has the transcallosal projection axon connecting to the other side of the brain. Now, in order to see whether this connection, this synapse is functional, which we did, we again transplanted neurons and then injected thalamus with the channel rhodopsin. I don't know whether you heard about the optogenetics. This is another very important tool in the brain research nowadays. So optogenetics are the channels which are sensitive to the lights. This is sodium channel rhodopsin, and when the light is exposed to this channel, to the cells, this channel opens, sodium gets in, and cell gets excited, stimulated. So what we did, we introduced into the thalamus these channels, and you can see in the green, this channel is labeled with the green. We labeled these neurons, and then lots of fibers came to the cortex. And then we cut the brain and put into the patch clamp system to record electrical activity of this tissue. And this one neuron, for instance, which we recorded, since it gets the synaptic connection from the thalamus, it has this channel rhodopsin. And when we recorded, it was neuron, but when we exposed light to this neuron, so the channels which are on the axon, they opened, the sodium got in, the action potentials were generated, and to our very big satisfaction, the recorded neurons get excited. So there was a direct synaptic connection from the thalamus to our grafted neurons. And that's the important part of the network reconstruction, that grafted neurons receive and this is the neuron we recorded. Look at this beautiful neuron, which are filled with the fluorescent substance after the recording. So that was very good news for us, that there is the functional connection from the rat neurons to our grafted neurons. And the grafted neurons, remember, this is human skin-derived neurons made into the cortical neurons and transplanted into the rat brain. So this is schematics, how it works. And then if you will record from this graft, you can also pick up the electrical activity due to the peripheral stimulation. If you stimulate paw with the nose or stimulate the paw, you can record the response from the recorded neuron. And this, the only way is that it is having the integration into the neuronal network, which is very important. Now, another part of this story is whether this only information coming from the host, can these grafted neurons send also information, make efferent connections with the host, with the rat brain, because that's another part, equally important part of network reconstruction, to receive connections and to send the connections. Okay, then we designed another study, and that's how it worked in the science. Every study gives you new questions, and then you design new studies to answer these questions. And we published a couple of years ago this in the American Journal Core Proceedings of the National Academy of Sciences, PNAS. And as you can see from the title, activity in the grafted human iPS cell-derived cortical neurons integrate into stroke-injured red brain, regulates motor behavior. So not only they make connections, but they regulate also behavior. Let's see what happened here. So we transplanted here in the cortex the cells, and then you see in the corpus callosum all these fibers, means that the transplant, since it's human, with the human specific antibody, you can detect all the human cells which are transplanted in the brain. And not only from the corpus callosum, they went to the another side, but they entered the cortex here, ectopic side, other side of the cortex, they entered. And that was good news. And we looked again with the electron microscopy and discovered that not only the afferent connection coming to our cells, but on the contralateral side, you can see the efferent connections. And efferent connections mean that this is the presynapse with vesicles making connections with the grafted cells. And this is the presynapse, and this is the postsynapse. And this is our grafted neuron, lots of vesicles you can see in the cell. And this is here the synaptic cleft. And then was quite obvious that we should continue digging more. What we did here in this study also, we did the reverse. In the previous study, I told you, we infected with the rabies virus grafted neurons. But here, since it's retrograde, we infected the host brain with the rabies virus and looked what's happening in the graft. So if the graft makes connections with the host, sends the axons and makes synapse, then we should be able to pick up the staining. When we infect the host with the rabies virus, we should pick up the rabies virus in the grafted neurons. So we infected first with the receptors and then put the rabies virus on the non-lesion contralateral side. And as you can see, this is the system here. If the host gets the rabies virus and our grafted neurons make synaptic connections, it will pick up the rabies virus and get stained. Okay, that's what is happening here. This is the rabies virus, this is the transplant, and to our big satisfaction, we picked up lots of stained cells in our transplant, meaning that our transplant makes efferent active connection with the host, not only receives connection, but also makes connection. And this is transcallosal. So there is no way in the world that there is any other non-specific staining or non-specific connection, because they are on the other side. One side of the brain, host brain, red brain, is stained with, infected with the rabies virus, and the transplant with its axons receives this rabies virus back and gets stained. Now, do they contribute also functionally to this events? And that's the last part of my talk. What we did here, we used again the optogenetics, but this time we used the halorhodopsin. And in the channel rhodopsin, it's a sodium channel and activates cells. Halorhodopsin is the chloride channel. And when you activate chloride channel in the neuron, you inhibit the neuron. So what we did, we introduced the channel rhodopsin, halorhodopsin, sorry, in our neurons before transplantation. So with the light, we can inhibit their activity. And that's what we did. We transplanted them. Now they have halorhodopsin. And if we will, during the behavior, apply the light through the cable, and you will see it shortly, to the rat, all the neurons which are transplanted, their activity will be stopped. And that's will allow us to see, if you will stop these neurons, their activity, what will happen with the behavior. And this is the cylinder test, when animal is put in the glass cylinder and it starts exploring surrounding and uses left and right paws to support. At the lesion on one side, contralateral paw is used less, and that's how you can assess the functionality of the paw. And here you see the rat exploring the cylinder. It has this wire which has the light, and you can see light is now on. which means that the inhibition of the neuron started. And what we discovered here, that with the transplantation, you can restore this damage here. If you will put the light, and if the cells do not have the channel halorhodopsin, nothing is happening. But if they have the channel rhodopsin and you put the light, the use of the pore is inhibited. And to our surprise, it was bilateral. So not only the contralateral side will stop using, but also both sides. And that's was very important discovery. And what was also very important, that 6 months after transplantation, when we took out the brain, made the slices and recorded in the patch clamp system, we discovered that transplant is still functional. With the depolarization, it can generate action potentials. And even more importantly, when you apply the light, it will still inhibit induced and the spontaneous activity. So this halorhodopsin is still active 6 months after the transplantation. So when what we observed with the light stimulation, it was real functional effect on the neurons. So by switching off the neurons, they use less pulse to support their body weight, which means that they contribute to the behavior. They become part of the normal neuronal network. And that's what our hypothesis is, and that's what we are currently trying to prove. So under normal condition, you have this bilateral communication from left to right cortex. It's called the interhemispheric inhibition. They are constantly inhibiting each other. When you get the damage here, you get the less inhibition here. Well, less inhibition means high excitation. So from contralateral side, you get higher excitation and impairment of the function. When you will put the, when you will put the transplant, then the transplant will restore, because it will make these connections on the other side. It will restore this balance and becomes the part of this interhemispheric interaction. But if you will block the transplant, then not only from transplant to the cortex contralateral, but also from contralateral will be impaired. And in the end, you will get both poles function impaired. So this is our hypothesis, which we are trying now to explore by making many, I mean, I will not go into detail. It's quite complicated, and it will take another lecture to tell you what we are currently doing. But what is important now as a take-home message, that neurons derived from reprogrammed human skin cells, so-called induced pluripotent stem cells or directly reprogrammed stem cells, can be used to restore stroke-damaged cortical network. And I hope I managed to convince you that this is doable. And with future work, we believe that we are not far away from the bench, from the clinic, but there is still a lot of work to be done. And I hope in coming years, we will do more in our research and get closer to the clinic. And this work, of course, it's not possible without team. This is teamwork. This is my lab. My former supervisor, Ulle Lindwall, who was whole my life, I worked with him, and he is still a senior professor. He's part of my lab. My colleagues and collaborators, Oliver Brustle from Bonn, who is one of the world leaders in generating the pluripotent stem cells. Karl Desserov, he is the one who discovered the optogenetics, and I guess he should get the credit. Jan Schobe, Molling Parmar, these are my local collaborators. And I'm proud and happy to say that all the electron microscopy studies, which I showed to you, they are done with my Ukrainian colleagues and collaborators and friends, Galina Skibo and Oleg Chupikov. And I am happy that despite the war, we still continue our work, and we currently submitted the paper to the journal, and they are actively involved in our work. And I hope when Russia will be beaten and world will prevail, we will even more extensively will continue our work with the Ukraine. And then all the foundations, of course, research is expensive and you need lots of money. And I'm grateful for all the foundations who provided money to perform this study. And I'm also grateful and thankful for you to listening to this lecture, and I will be happy to answer a couple of questions.
Thank you, Badnazar. If there are any questions, please, you can raise the hands and then we allow you to ask the question. Please.
Excuse me, sir.
Yes, go ahead.
So can you hear me?
Yes, I can.
Yes, please, please.
Okay. Sir, I had a doubt, like you told us that the transplant, like the stem cell transplant, but how is it done? Like it is, these cells are taken, these skin cells are taken from the host or the the donor, which should be a match or?
Well, we haven't done it in humans, and this is the human donor-derived skin-derived stem cells, and it's done in the xenotransplantation setting, so human cells into the rats, in order to avoid the rejection of these neurons, because you know that from one species you can't transplant. We take the special breed of the rats, which lacks the T cells, so there is no rejection. You can do also with the immunosuppression, the xenotransplantation, but normally we work with this red line, which called nude rats without T cells, and there is no rejection. So cells are coming from the human donor, and the recipient host is the rat, nude rats.
Okay, thank you, sir.
You're welcome.
So
there is
please,
Subhanshu,
please.
Go ahead.
Sir, you said that these neuronal connections work completely with the hosts, right? So yes. I have a question that can these transplanted neurons be somehow regenerated and can be used to cure the demyelination diseases?
Excellent question. That's what I told you. We just submitted the paper. That's what we studied on the oligodendrocyte production from the same cells. That, you know, with the stroke lesion, you have demyelinization, oligodendrocytes, lots of oligodendrocytes die. And we discovered that our stem cells can generate not only neurons, but also oligodendrocytes. And currently we have, actually yesterday we received some reviewers' comments, which we need to address, that we can myelinate the axons by transplanting these cells. And that's very important because without myelin, there is no proper connectivity, and you need to have the myelin, which is produced by oligodendrocytes. And with stroke, not only neurons, but oligodendrocytes also die, and there is lots of demyelination ongoing, and you need to restore not only the connections, but also you need to myelinate these demyelinated axons. That's what is happening right now. Thank you for a very good question.
Thanks a lot, sir.
Shundashu, you can ask the question.
Ma'am, I already did.
Please, if there are any more questions, I can take one or two questions.
Hello, thank you very much.
Anjali, go ahead.
Good afternoon, sir.
Good
So
afternoon.
can we use this technique to like regrow the nerves for the patients who have like nerve damages?
Regrow what? I couldn't hear you.
The neuronal cells, like
Yes.
for the patient who are having the like nerve compression or the like,
Oh, yeah.
nerve damage.
Yeah,
Can we do
this is with the peripheral nerves. I mean, there is lots of ongoing study. We are not working with it, but there is lots of ongoing studies to regenerate the nerves, and definitely it's possible. It's not in the clinic yet, but what I can tell you as some extra bonus, that next month they will be in our clinic here in Lund, in Sweden. We will start the stroke not stroke, but Parkinson patients. So Parkinson patients will be transplanted with the embryonic stem cell derived dopaminergic neurons. And that's the milestone, because previously we were doing it with the fetus derived, aborted fetal dopaminergic neurons. But now we are ready and we have all the permits and everything to start with the patients, Parkinsonian patients, and replace dead dopaminergic neurons with embryonic stem cell derived dopaminergic neurons. And I think this is the future of the brain regeneration clinics. And I hope at a certain point we will be able to do the same with the stroke patients, what we are starting now with the Parkinson patients.
As I have information, there is another question from conference hall, and we can allow them to ask
That
the
should
question.
be the last question.
Hello,
Yes.
thank you very much for a very interesting lecture. I have two questions. Can this technology lead to tumor formation in the brain? Did you have such cases in your laboratory practice? And my second question is, how do you think when this technology can be applied in clinical practice, like prognosis? Is it 10 years or I don't know, 1 year?
Yeah, when it comes to the tumor and the cancer, these cells are for the regeneration. Of course, if you tumor post-surgically, if you remove tumor and you have the brain damage, of course, you can consider the possibility to use the similar approach. When it comes to the tumor treatment, these type of cells can be used as a vehicle for delivering certain molecules or certain drugs towards the tumor, and to kind of be used as a killers, not for the reconstruction of the neurons, but but this is totally different approach and for totally different scenario. That's feasible, of course. And then when it comes to the time frame, it could be somewhere between 5 and 10 years, I hope, because there are lots of procedures which we are already starting to do it suitable and accessible for the clinical setting, lots of safety, lots of derivation protocols, etc., etc. So lots of work to be done before we reach the clinic and get any approval for the clinical trials.
Thank you. Sorry, about tumors. I meant if you are treating a patient with ischemic stroke, for example, and with this technology, might it lead to tumor like?
Okay. Sorry, I misunderstood. I'm sorry. No, this is a very important question. Of course, we have done lots of tests. I mean, imagine these rats are the T cell deficient, and T cells are the main killers of the tumor cells. If there is any chance of getting tumor, this is the chance today to get it in the T cell deficient animals, and we've never seen any tumor formation. So they are quite safe, and they are not pluripotent at the point when we are grafting. All they can produce are the neurons and oligodendrocytes. There are no other cells which they are producing. And we have long observation, like 6-8 months after transplantation. If there would have been any tumorigenicity, that would have been revealed already, and we haven't seen it in any single case.
Thank you. And these cells, they should be derived especially from the same patient, or there is patient one?
It will be very difficult to do it with the autologous transplantation. The idea will be that it is allotransplantation. We create the bank of the cells, and then with initial immunosuppression, you can make them to survive. And you know that the brain is immune privileged, so. And that has been done with allotransplantation with the Parkinsonian patients, and we know that it will work without autologous. Autologous will be very time-consuming and very expensive, so it's not realistic that you can do it in an autologous way, but allotransplantation is quite realistic.
Thank you very much.